Wow. Indeed, seed is life and life is seed. Seeds also form the basis of many things. Many medicines come from seeds.
I'd like to state that if you look at plants, plants give medicines, help with the conversion of carbon dioxide to oxygen, which gives life, which contributes to the continuation of life. Originally, medicines came from plants, from roots, from barks, but they all came from seeds. Then they were copied scientifically and made into synthetic medicine.
And many seeds then have a fruit around them. Think of a tomato. And that comes from the mummy's wall, the tummy. That's the ovary wall, which then develops and becomes a fruit. And that often has a dispersal function.
Although the produce of many annual crops like garden beans, cucumber, pumpkin and tomato are actual fruits, such fruits are regarded as vegetables by consumers, together with true vegetables like beetroot, cabbage, carrot, lettuce and onions. The enzymes in raw fruit and vegetables assist with the digestive process, and the body will subsequently have more energy. In other words, eat more raw fruits and vegetables for more energy. The 5-a-day movement maintains that if the only change people made to their lifestyles were to eat at least 5 servings of fresh fruits and vegetables each day, cancer rates could drop by as much as 20%.
Seed is life, life is seed.
The modern cultivated tomato is well known for its ability to combat certain forms of cancer. Plant improvement through conventional or traditional breeding has contributed significantly towards these favorable attributes.
Our whole purpose and aim is to breed tomato varieties for the market. The breeding that we're doing is more a traditional type of breeding, where we would make a cross and let the population segregate, and selecting the best lines out of those segregating populations. At the end of the day, we will then get to an inbred line, and those 2 inbred lines we would cross together in order to give us what the market requires. The whole idea of making a new hybrid is to combine specific characteristics that would be of benefit to the consumer or the farmer. In this case, we've got the pollen from the male plant, and we need to transfer that pollen onto the female plant. In this case, this female plant would have a specific set of disease resistances, and the male plant would have another set of disease resistances. And we're going to cross these 2 together in order to give us a hybrid that would have all of these benefits together. So first of all, before we make the cross, we need to emasculate the female flowers. But we're going to emasculate it by actually taking away the anthers here. As you know, a tomato is a self-pollinate plant, and because of that, we need to take away the males or the anthers approximately 2 days before the flowers are open, like that. After we've emasculated the flower, approximately a day later, we would collect pollen from the males, and normally we would collect pollen in a tube like this, and then we would transfer the pollen onto the stigma of the flower. And we would do it in the following manner, just softly dipping the stigma inside of the pollen. And there it is, pollination done. Approximately 2 to 3 weeks later, you're going to see lovely fruits like this that have developed. These fruits would be ready in about 3 to 4 weeks from now. We normally would pick the red ripe fruits, and then we would take the seed out, clean the seed, treat the seed, and then that's the seed that's the new hybrid, which can be transplanted in order to give us that specific hybrid. There are different types of tomato varieties that we would breed. We've shown you the varieties that's suitable for production under protection. In this specific field, we've transplanted the hybrids that's more suitable for field production. And again, we would be looking at these hybrids and evaluate these hybrids to see that the quality aspects are those like firmness, long shelf life, color up, disease resistance, and all of those things that would be important not only for the grower, but also for the consumer. We would be breeding tomato varieties for different markets, as mentioned, and I've shown you the varieties that we're going to evaluate under protection. In this field here, we will be transplanting the varieties that's more suitable for open field production like this. And again, in this field, we would be looking at the different quality characteristics suitable for that specific market. And disease resistance is very important when transplanting in a field. All of those things we will take into consideration before we make a selection for a specific hybrid for the marketplace.
We are the Quality Assurance Lab.
Although genetic considerations are extremely important for yield improvement, farmers will not benefit from these advantages if a seed lot has been contaminated with pests or seed-borne diseases, weed seed, or other materials such as stones, chaff, etc. Germination capacity, of course, is also crucial. Most seed houses in South Africa have seed testing laboratories, registered and monitored in accordance with national legislation. All tests conducted are standardized according to the rules and guidelines prescribed by the International Seed Testing Association. National legislation prescribes minimum standards and labeling requirements with which all seed merchants in South Africa must comply. This is an example of the germination test on tomato seed. A counting frame is used to plant 4 replicates of 100 seeds each. A specially manufactured roll paper medium is used for these tests. Germination capacity is monitored in a specialized germination cabinet capable of simulating night and day cycles at 20 to 30 degrees Celsius alternating temperatures. The tomato originated in the Andes Mountains of Peru, from where it spread extensively to South and Central America before being introduced to the rest of the world. Although it was first used as a vegetable comparatively recently, its consumption has increased so rapidly that in most countries of the world, it can now be described as the most important vegetable.
Seed is life, life is seed.
The common bean is divided into garden beans, which are utilized as a green vegetable, and dry beans, which are regarded as a field crop and produced in bulk for processed food products, like canned beans and soups. The bean and its relatives like ground nut produce relatively big seeds with their embryos already well developed within the seed, in comparison with cereal crop seeds, for example. They are subsequently extremely vulnerable to mechanical damage, and special care has to be taken during harvesting and cleaning operations for seed production.
Here we are now at our seed processing facility where we clean our seed. At the moment we are busy processing garden bean seed. Upon seed receipt, samples are drawn and submitted to our laboratory on site. There it is tested for its physical purity and also for germination. This is necessary for us to predetermine the quality of our seed lots so as to know how to set up our machines to obtain maximum seed recovery. Here we have an example of the finally cleaned product after it's gone through the pre-cleaning phases where the sticks and pods have been scalped, as well as the finer inert matter like pods and leaves have been separated by the screening process, and then also where all the defects have been removed by means of color sorting over here.
After cleaning and prior to packaging, proper seed quality control is indispensable. This is of particular importance in the case of bean seed, as mechanical damage can be lethal to seed or result in various seedling abnormalities that will prevent seedling development into a mature, productive plant. A grading test on samples received from the seed cleaning plant is conducted in the seed testing laboratory. Here, fractions of mechanically damaged seed, discolored seed and seeds of other crops or species are, for example, separated from the actual clean seed in order to monitor the efficiency of the seed cleaning plant for this purpose. The germination test is usually conducted in specially prepared, sterilized silica sand.
I've got a container. The substratum is sand, and inside this, 6 days ago, we planted beans, just to prove to you how a live seed really is. Now this is ready to be evaluated. We usually plant 4 replicates of 100 seeds to give us 400, just to get the statistics right. And just to show you how we evaluate this, each one of these seedlings are taken out of the sand, and all the structures are now checked to see whether it's a normal seedling and whether it will develop into a healthy producing plant.
Beans, indeed, constitute a truly natural food for the health-conscious public of today.
Seed is life, life is seed.